Hi, my name is Alexandra DePinto, and today I will be reporting my Bio 282 news story. The news story that I chose relates to the topic we learned this semester, protein structures. Proteins are an essential component for all living things to function. They replicate DNA, help move other molecules, respond to signals, make reactions happen more quickly, and etc. With the present pandemic of SARS-CoV-2, I chose this news story. The coronavirus mutation has taken over the world. Scientists are trying to understand why. It incorporates the knowledge I learned about proteins and the information on SARS-CoV-2 spike proteins. Here's a little background information on COVID-19. COVID-19 was found in the first human in Wuhan, China in December 2019. It is commonly found in animals such as camels, cattle, cats, and bats. However, it is very rare for the coronavirus to infect people and spread between humans. There are two other known coronavirus strains that have occurred before SARS-CoV-2, which include MERS-CoV and SARS-CoV. All three viruses have origins coming from bats and are known to cause severe upper respiratory illness in people. With COVID-19, however, it spreads much faster and is causing mild to severe illnesses of respiratory problems, which has led to death. This makes this coronavirus much more concerning than the other two. The news story being reported today addresses the world's and scientists' concerns and interests on the new mutation of COVID-19. It can be seen from the image above that the mutation is occurring at the spike protein. What is the spike protein, you might ask? Well, it is the protruding structure that gives the coronavirus its crown-like appearance and mediates the host cell binding and entry of the virus. Since the first appearance of COVID-19, researchers have discovered that about 70% carry the new mutation called D614G. On a microscopic level, a mutation in the spike protein of SARS-CoV-2 may only change one amino acid out of 1,300. However, this one difference may make a difference in how the virus attacks human cells. Since the new mutation has left scientists scrambled to learn more, there are many lab experiments being conducted to learn more about this mutation. From recent lab experiments, scientists have found that the mutation makes the virus more infectious. A study done at Los Alamos National Laboratory found that patients with the G variant actually have more viruses in their body, which is why it makes it much easier to spread. However, patients do not get sicker. A suggested hypothesis for the reason why D614G mutation is much more infectious is that the mutation allows the spike protein to change shape as it attaches to the ACE2 receptor, improving its ability to fuse to the host cell. Although the hypothesis and experiments are compelling, they are not conclusive. However, the mutation's differences are found in the spike protein, which scientists know play a huge role in human cells. This article, originally posted by The Cell, is called Tracking Changes in SARS-CoV-2 Spike, Evidence that D614G increases infectivity of the COVID-19 virus. Confirms this news story. In this study, Krober, along with other authors, present recent evidence, as can be seen in the figures above, that the original SARS-CoV-2 spike protein, which mediates the host cell binding and the entry of the virus, found in the first human case in Wuhan, China, called D614, has changed patterns towards an increase in the G614 variant. The G614 variant is slightly different from the original D614 spike protein in that the G614 is associated with higher levels of viral nucleic acid in the upper respiratory tract in humans, suggesting higher viral loads and higher infectivity. This article further scientifically explains what the news story was conveying. Although the hypothesis of the spike protein changing shape as it attaches to the ACE2 receptor was not illustrated, the article confirms the higher contagious rate of the mutation as discussed in the news story because of the mutation of the spike protein. Thank you for listening.